or you can send me an email at angel.sampson@ufl.edu. So if you have any questions, you want to see Wes, you want to plan an experiment, please reach out. I'm here to help. And we've invited Greg here. He's the field application scientist for our region, and he's an excellent source of information, and he can also help with with getting the reagents, and also if we run into any problems with your assay to troubleshoot. So with that, I'm just going to go ahead and transfer over to Greg. Thanks for coming today.
Thank you so much for that introduction. Now, I do want to say that you at University of Florida do have an amazing resource in Angel. She is a wonderful wealth of knowledge. I've had the opportunity to work with her, working with some other people as well. So it certainly is an advantage that not many people have. So I would encourage you all to reach out to her as well as to me. But you all have that wonderful resource on site, so you all are very lucky. But as Angel said, I am your field application scientist. I live in Durham, North Carolina, but I certainly come down to Florida often pre-COVID, and hopefully now as things are starting to look a little better, I'll be coming to visit y'all in person. But anytime you want to set up a Teams meeting or a Zoom meeting to discuss your data, I'm always happy to do so. On the call as well is your Simple Western sales rep, Lynette Grove. She is here to help you all with any questions about ordering reagents, figuring out what you need, where you need it to get it, that sort of thing. Lynette is awesome with that. Not only that, thanks to some constraints, we're feeling the same supply chain constraints that everyone else is feeling. If you are ever in a situation where you need reagents and need them fast, we are your advocates here. And especially Lynette is very good at finding things somewhere in the company. Lynette, do you want to just say hi?
I will just say hi. Thank you so much, Greg. And I'm going to hand it back over to you, and anything you need, I'm always here.
Yeah. We do work as a team, so if you're ever wondering who to contact, we talk to each other all the time. So if you send a question that's really more for me or for her, we'll forward it between each other. But anyways, just to talk a little bit about what we're going to cover today, you have a Wes in your core facility that is available to all of you. And I would like to give you some information today about what the Wes is, how the Wes works, and then some things that you need to consider for getting started. And just a basic primer of like, okay, what do I even do with this? And how do I get to a point where we're at a functional assay? And of course, afterwards, after you've learned all this information, I do encourage all of you to reach out to me. I'd be happy to set up a 30-minute or hour-long consultation about your science and what you could potentially do with Wes to help you along. But yeah, just to get started, we are part of Bio-Techne. So Bio-Techne is a large company that has multiple different brands associated with it. Lynette and I are specifically part of the ProteinSimple brand. And so the ProteinSimple brand of Bio-Techne is really the instrumentation arm for protein characterization. We have a lot of other brands with us that, like most people recognize R&D Systems as one of our largest brands. They are for the quantitation and chemokine kits and recombinant proteins, antibodies, that sort of thing. We also have Novus Biologicals, so another antibody company. But really for ProteinSimple, we are the instrumentation portion. And if you look at the instruments that ProteinSimple has made over the course of our entire existence, our instruments have been all about detecting proteins in a capillary and characterizing what is going on with these proteins in a capillary. We started with instruments that did isoelectric focusing, which was really for manufacturing, to make sure the proteins that were made were folded correctly. And then we got really good at working with proteins in a capillary, figured out that we could cross-link those proteins and do an immunoassay on those proteins. And so that's really where the simple Western systems, and Wes in particular, is going to be. And so when we consider what does Wes do? Well, Wes is not going to blot proteins to a piece of paper. It is not a Western blot at all. It is a Western blot replacement or a next generation of Western. And it really comes down to what is wrong with a Western and why do we need to even replace it at all? And that's, these are not from my dissertation, but they certainly could have been. These Western blots, I certainly have a lot of experience doing traditional Westerns. And there's the good and the bad and the ugly of doing these multi-step Western blot experiments. And if we actually look at just the process, from pouring gels all the way to the results, you guys might not be pouring gels. That might be something that some of us older folk had a lot more experience with. I know a lot of folks, I would like to say spoiled, that you can buy your precast gels, but certainly all these different steps are manual steps that could introduce error. They could introduce variability. And so any sort of that variability leads you to question, okay, is the result I'm seeing actually a result? Is this something that is happening in biology? Or is this just something that, is actually from my process? So what Wes does is it takes the human element out of a lot of the processing steps. So instead of, loading a gel and doing all these different things to make sure that your gel is appropriate, working fine and all that, with Wes, what we'll do is we'll prepare our lysates almost exactly the same way we would for a traditional Western. Instead of the blue 4X Lamley buffer, we're going to actually give you a loading buffer that's designed for our system. But other than that, for the most part, any lysate will be able to be transferred over. We do have a buffer compatibility table that I can send to you where we can check your lysis buffer to make sure there aren't any components that will interfere. But again, for the most part, most of our lysates transfer over directly. And all we're doing is putting them into a different loading buffer. And so we'll prepare our reagents, load them into a Wes plate. And so this is a consumable that we will send to you. These wells are kind of the size of a 384 well plate. So if you've loaded a lot of 384 well plates in your lifetime, this is going to be pretty easy. If you have not, it takes a while to get the muscle memory, but once you get it, it's pretty straightforward. My record for loading a plate is 22 minutes. And that was, I don't think I'll be beating that record anytime soon, but typically for loading a plate, gonna be anywhere, well, first time you do it about 2 hours, probably a good idea to buffer or plan that time. But once you get going, 40, 30 minutes of hands-on time, loading the plate, similar to like what you would do when you're loading a gel. But once this plate is loaded, you're going to put it in the instrument, put the capillary cartridge in the instrument, close the door, hit start, and walk away. And 3 hours later, your experiment is done. So there's no going back and doing washes and other things like that. So certainly saves you a lot of hands-on time. And really all you're doing is in this plate, you're loading your molecular weight marker, your sample, your blocking buffer, your primary antibody, your secondary antibody, and luminol peroxide. So all these reagents are put in the plate, put in the instrument, and done. So exactly what is happening when you load the plate? Well, once you put the plate in the instrument, the Wes will put the capillaries into a separation and stacking matrix. So this separation and stacking matrix is located at the bottom of the plate. Wes will put those capillaries in, apply a vacuum, and build the gel within each capillary. Now, it's important to think about when we're thinking about how are we going to set up this experiment and what is it going to mean? Every single capillary is going to be its own independent experiment. So with traditional Western, sometimes you have to watch out, well 1 might, if you overload it too much, interfere with well 2 or well 3, and you might have bleed over. But in this case, each capillary is completely independent. So you don't have to worry about well 1 interfering with well 2 or well 3. Now, once we have our gel equivalent made within the capillaries will go into your sample and aspirate up about 1/10 the length of the capillary with sample. And this is something that we can modify when we go into advanced troubleshooting. We can change how much sample we pull up. But if we think about in a traditional Western, when we load our sample into the well, we're loading a certain mass. So we're loading 15 micrograms in one well, micrograms in another. And it's based off the amount of volume and then the concentration of that volume. Now for Wes, when we're loading the plate, we can load 3 microliters of sample in one well, 5 microliters in another, 10 microliters in another. Wes is going to take the same amount in every single capillary. So it doesn't really matter how much sample we load, as long as we're loading the minimum, which is 3 microliters, so the capillaries can get in the sample. But the capillaries are only going to pull up 1/10 the length of the capillary, which is about 40 nanoliters of your sample, a very small amount. Now this, when we're thinking about transitioning from traditional Western to this new methodology for doing an immunoassay, traditional Western, we talk about how many micrograms did you load in the well. That's the important number to discuss. For simple Western, it's about the concentration of the sample that we put in the well. And typical sample concentrations are going to be usually around 1 mg per mil or 1 microgram per microliter, depending on which you want to say. They're the exact same thing. But for simple Western, typically somewhere around 1 mg per mil is the concentration that you're going to put in. And you're going to put in 3 microliters of that. So 3 micrograms of your cell lysate, typically. Going to save a lot on lysate, using usually about 1/10 of what you would for a traditional Western. But once Wes goes into that sample well and aspirates your sample, we apply a voltage, same thing you would for a traditional Western, and separate out the proteins based off their apparent molecular weights. Now at this point, Wes will shine a UV light on these proteins. The capillary inner wall is coated with a reactive chemical that will cross-link to your proteins and bind them to the inner wall of the capillary. So now your proteins are stuck. They're on the capillary, they're not going anywhere. You have denatured proteins stuck to the inner wall of the capillary. Then we can flush out all of your sample, everything that didn't bind, all the separation and sieving matrix, all that stuff gets just washed right out of the capillary. Then it's a question of what we do with that sample. We can label with a primary and secondary antibody and detect with HRP and quantify that. So basically doing what you would with a membrane, incubating with that primary antibody, then incubating with the secondary antibody. All that is automated, done within Wes. We don't have any variability of how much sample we loaded into the capillary, because Wes is loading the same amount of sample into every single well. And so what does this mean as far as data? And so we had this experiment that was done in San Jose, where we were looking at ERK1 levels and HeLa lysate. And we had 4 people. These are not pictures of the people, these are stock photos. But we had 4 people at our internal office run an assay looking at 11 replicates of the same lysate, detecting ERK1. First one had a CD of 5.2%, 11%, 12%, so on and so forth. But the cool thing about this was 4 different people on 4 different days taking the same lysate, they got the same answer. Same level, same amount of signal. CV of less than 10% between all 4 of them. If you think about this from a perspective of working in a lab, I know for me during my PhD and postdoc, there was a lot of variability between user and user for doing a traditional Western. Yeah, and there'd be the one postdoc who is the only one that could get that protein to work. And certainly it can be frustrating when that person leaves the lab. And so this automated system really does help with the reproducibility. So you can have really good reproducibility from person to person and from site to site. When we talk about our understanding and being able to make decisions based off, is this change biology or is this change based off our process? We can have a lot more confidence that it is biology. And this is just another example, taking the same lysate, running it all the way across the capillary for CDK6 and PI3 kinase, really good reproducibility across 24 different capillaries. And so when we're planning our experiment on Wes, there are two different capillary cartridges we can use. One is 13 capillaries, one is 25 capillaries. And that gives us, because we have one molecular weight marker, we have the option of basically running a 12-lane gel or a 24-lane gel. Most people do the 24-lane gel. The reason is, if you want to look at a different antibody, you don't have to cut up strips or things like that you would for a traditional Western. You just load that different antibody into the wells for the primary antibody. So antibody screening becomes very, very easy. In theory, you could test 24 different primary antibodies all in one capillary cartridge. Of course, you don't have any negative controls, that's only positives. So as a scientist, you need a negative control. So really more like screening 12 antibodies all at once. But certainly gives you a lot of flexibility in your detection and building your assays. So when we talk about how we're going to display this data, we are not blotting proteins. So traditional Western, you're blotting a protein to a paper. And we can actually show your data from Simple Western in a view that looks like a traditional Western. We have some really cool tools for that. But the data that we're getting, we're separating out proteins in a capillary. So what does that sound like? What kind of experiment does it sound like when you're separating out proteins in a capillary? The data, the raw data we get, is most analogous to an HPLC. And so we get this HPLC-like trace, the electropherogram. Now, this gives us a really good way to visualize the data and show the data in the most data-rich methodology. But we'll give you both views of the data. And most people are actually publishing the lane view when they publish it. But when we do our quantification, we will do an area under the curve, similar to what we would do for HPLC. And that's going to be automatically reported by Compass. So when you get the data, you will see several different peaks all the way across the capillary. And all of these peaks will automatically be quantified by the software. So really fast ways to generate quantification and start to interrogate what's going on in your samples. And the Simple Western technology has actually been around for a very long time. And so over a decade, and we have a lot of publications out there. So this isn't just, something brand new. We aren't the new kid on the block anymore. We've been around, we've been established. And I can talk till I'm blue in the face of data from San Jose, where our company is headquartered, but it has been published as well. And there are some really cool publications out there for all of you to check out. I'm happy to discuss with you about them further. But this is just one example of folks looking at ERK2 levels across multiple different cell types and multiple different runs, and they see really good precision. This is not from ProteinSimple, this is from the NIH. So this technology, again, not new, not the new kid anymore, definitely established and trusted by multiple people. One of the places we're really seeing this technology take off is in the gene therapy space. So a lot of gene therapy companies are actually relying on the reproducibility of the system. I've worked with a couple of companies now that have done IND submissions using simple Western assays for validation of the efficacy of their gene therapies. So definitely a lot of cool things going on in the simple Western space. And as I mentioned, there are a lot of cool ways to show your data. One of the new things that we have in our latest version of our software, Compass 6.0, are these lane annotation tools where we can actually very quickly make publication quality figures within the software itself. So it saves a lot of time, easy ways to keep track of and curate your data. One thing to note about our software, Compass for Simple Western, is that it's free for anyone to download. There are no limitations or restrictions. So anyone who wants to use the software is perfectly welcome to do so. And I'm happy to help you out with using the software and learning the software package. But really the big thing is huge time savings. 30 minutes is very generous. This is a nice marketing slide where like you can do it in 30 minutes. The first experiment you do is probably going to take you 2 hours. But as you get that muscle memory, as you learn where you need to put things, this will drop in time. So really hands-on time really drops, gives you a lot of time. You don't have to every 10 minutes go back and change the wash buffer on your membranes, things like that. So frees up your day a lot to do other things and focus on moving your projects forward. And so really, if we're talking about this, Jess, Wes, and Abby, 30 minutes, 3 hours to results. We have people who will crank on these instruments and set up 3 runs a day. So 1 in the morning, 1 in the afternoon, and then 1 that's running overnight. It has been extremely popular during COVID because when people were working shifts, fitting in a traditional Western into short shifts was very difficult, but a simple Western is 30 minutes to 45 minutes of hands-on time. So it really opened up a lot of ability for people to get their data, even if they were working a short shift or trying to limit the number of people in the lab. But one of the cool things about this, again, you're going to load 3 microliters of your sample into every single well. So this saves a lot on sample. And again, with traditional Western, you're talking 20 to 50 microliters per lane, so 20 to 50 micrograms of protein, depending on what protein you are looking at and what the abundance is. We can change the concentration of our lysate, and we can load higher concentrations of our lysate to get more signal. We can aspirate more sample into the capillary if need be. That's really more of a second step that we would take to increase our signal. So we're not locked in, we're not limited, but certainly we do save a lot on the amount of sample that we load. And this is just an example, from Germany. Dr. Reiger was looking at mouse hematopoietic stem cells and running into an issue. If he were to isolate stem cells and run them on traditional Western, it would take 50,000 cells, which is 50 mice. So about several $1,000s per lane to do this on a traditional Western. So it was cost prohibitive to do immunoassays on these mouse hematopoietic stem cells. But simple Western really cut the cost of this down and enabled him to actually interrogate what was going on in these cells. So thinking about your experiment, if you are culturing cells in 10 centimeter or 15 centimeter dishes, you're probably not worried about the amount of sample that you're using. But if you're starting to do different discrete cell types or different tissues using mice, any of those other things, or if you're going to ever transition to those, certainly keeping in mind that you can save a lot on sample, run a lot more tests per each of one of those samples. It certainly can be advantageous if you are in a sample limited situation. And when we're talking about that quantification, bringing down the variability does help you when you are looking at more quantitative methodologies. And the most relatable methodology for this is an ELISA. And so people use ELISA to do relative quantification and try and get quantification from the immunoassay. And ELISAs are great. They work really well for well-qualified antibodies, but you don't have the size separation, so you can't see any post-translational modifications. You really can't see any processing of the protein. And if the antibody you're using is not very specific, you could not really get a very reliable ELISA. And so same advantage that you get for Western over ELISA, you're going to get for Simple Western. You're going to be separating out these proteins by their size. And we do have people running a standard curve of samples and then doing absolute quantification of the protein within their samples. And that is something that actually the software can set up. So it can do a linear or quadratic fit for your samples, depending on what you want. Or you could just take these numbers and put them into Excel if you wanted to do that manually. But the software can do it automatically for you as well. And so that's another really cool thing that we do have more and more people using and taking advantage of. And that's just an example right here, the standard curve. We got the slope, the R-squared of 0.981. So this was a pretty good example of using Simple Western to make a standard curve and then do absolute quantification. And as I mentioned, we've had a lot of people use this, especially in the pharmaceutical space. This is a published example from a group in 2015. So, this has been true, tested, published experimental methodology. This was a biotech company just looking at the quantification of the drug substance they were making. And they were seeing, again, great linearity with this system and very nice precision. So this is why you would use Simple Western, because it's going to save you a lot on sample, save you a lot on time, and just give you a lot more confidence in the data that you are producing. But before I go into how you make that transition, I'd like to pause for a second. Are there any questions about how the system works and why you would use it? Well, looks like nobody's taken themselves off mute. So,
hi, Greg.
Yeah,
I'm Hugh McDowell. We're working with a very, very low concentration of protein. It's in the retinal tissues. I mean, so low I can't even guess how much of the protein is in there. Are there any tricks that should happen? I mean, we also have some other problems with it too. Antibodies aren't terrific for this particular protein, but is that something that you think would work well on this system?
So I've worked with a couple of labs at down in Miami, as well as here in the Triangle, working with single retina and doing single retina detection of a couple of proteins. As far as detecting those, they've been successful detecting their proteins. Immunoassays on Wes suffer the same challenge that a traditional Western does, that you need a good antibody. And really the advantage of Wes is that you could screen multiple antibodies and find the good one fast. But if your antibody is not good, then unfortunately, there's no magic box in the world that's going to make a bad antibody a good antibody. But we have had people with very small amounts of protein be successful on the Wes. And that's if you have very low concentrations, certainly, making the lysis of those proteins in as concentrated as you can get And then we can make changes in the way that JESS pulls the sample into the capillary and just pull more sample in. And we can certainly do a lot of things to try and see if that will work. It will ultimately depend, on the expression of the protein and the quality of the primary.
Yeah. Do you know, I'm hearing this as if this is a lot more sensitive than traditional Western. Do you have a number for that? Some guesstimate of how much more sensitive it is?
So compared to traditional Western, in traditional Western, we can load more concentration of the protein. We can load 50 micrograms, whereas, if we're talking about absolute detection of the protein, we are going to be a lot more sensitive. But when we're talking about concentration, we're a little more sensitive. And it's hard to do a direct, like we're 10-fold or we're 20-fold, because every case is different. Every antibody is different. And so, I would love to give you a solid answer, but at the end of the day, we have to do the experiment to see whether or not we're going to see better sensitivity on the system with your samples than we would on a traditional.
Okay, thank you very much.
Yeah. All right, thank you for your question. Are there any others? All right. Well, if there are any more questions, please do feel free to take yourself off mute at any time and let me know. And I am also happy to have a discussion with you at the end of the presentation. If you want to send me an email, we can set up an individual time to talk. But when we're starting and talking about that transition, like how do we test things on Wes and how do we start to transfer our experiments, there are some important things to think about in the difference between how a traditional Western is run and how a simple Western is run, and what that means as far as what we're going to load into the wells to get a successful run. Now, of course, when we're starting an experiment on the Wes, the big knobs that we're going to turn to make our experiment work, same things you would on a traditional Western. Primary antibody, what is that primary? Is it good? Is it bad? Does it have a lot of non-specific signal? Finding that good primary is going to be key. And how do we use it? What concentration should we use for this primary? Are we using 1 to 1,000? Are we using 1 to 10,000? On traditional Western? And how does that translate to simple Western? And lastly, how much sample do we load? Are we loading 1 mg per mil? Are we loading 3 mg per mil? Are we in a point where we can't get a concentration of our sample, and so we're just loading everything that we can? These are important things to think about when we're developing a simple Western. And when we're thinking about what we want to achieve, it's important to understand what questions we're asking. Are we trying to be quantitative? Do we have 500 samples and we want to tell what's going on with this protein in 500 different tissue lysins? Are we really more trying to be qualitative? We have a protein and we have a knockout. We want to confirm that that knockout worked. So we're looking yes or no. Is that more qualitative? Yeah, the level of energy that we put into figuring out how to get our detection working is going to result in how quantitative we are getting our detection. So there's the old adage in science, garbage in, garbage out. You have a garbage primary. If you have a garbage setup for your experiment, you can't expect absolute quantification of your protein. But if you put in the effort and put in the work and really refine your assay and get your assay solid, you can get really good CDs and really good reproducibility with the Wes. But it all depends on what you're trying to achieve. If you've got a bank of samples and you want to know, okay, do I want to look at this protein? And I'm not even sure if this protein is going to vary in my samples. You might want to look at it with a qualitative assay just to screen at first, and then take that to a quantitative assay and put the energy into developing it into quantitative before you really dive through your entire sample set. But one thing that's important and one thing that's going to be different from traditional Western. In traditional Western, we have a membrane. It is soaking in a tray of our antibody. All of the membrane gets the exact same antibody solution. In simple Western, every single capillary is independent and gets its own independent primary antibody mixture. There's no sloshing back and forth from well to well. It's all one mixture. Because of that, if we have any evaporation over the course of waiting, from when we put our plate in to when that antibody solution is getting into the capillary, that's going to change the concentration of the primary antibody in specific wells. So how do we account for that? How do we build robustness into our system so any of that variability doesn't affect the outcome of our assay? And when we're trying to be quantitative, we want to build as much robustness as possible so that any of those changes, if we make up our antibody solution and we make it a little differently, we don't vortex it very well, and so different concentrations are going into every well, we don't want that to have an impact on our output signal. And so the way we get around this and the way we build that into our system is by using our primary antibodies in saturating concentrations. We want to have so much primary in there that any of the available epitopes are bound up by our primary. And that's just depicted right here. So on the x-axis, we have our antibody dilution, and on the y-axis, we have the output signal. So if we are in a situation like we are right here, and we have not vortexed our antibody very well, and in well 1, we have an antibody concentration that's about 1 to 250, and here we have an antibody concentration that's maybe 1 to 100. We're going to see very different signals from our experiment. We're going to see at least a twofold change in the signal. But if we're in a situation like we are right here, where all the potential epitopes are bound up by antibody, if we have a evaporation that evaporates half the well for some ridiculous reason, I mean, you guys are in Gainesville, humidity's high, I wouldn't expect evaporation to be an issue. But if for some reason it was a very dry day and all the stars aligned in a very weird way, and we had that twofold concentration of our antibody, we have no change in our output signal because we're already at a point where we are saturating all the epitopes. So that is something that when we try to be quantitative on a simple Western, our goal is to find an antibody that can be used in this way. This really is just taking advantage of the system. If we want the kind of data that we were getting from a traditional Western and we just want to transfer over what we're getting, we can be more qualitative. We don't have to worry about this so much. But if we're really trying to be quantitative, we need to take out all the potential variables that could exist. And by using our antibody in saturating concentration, we're going to give ourselves that reproducibility day to day. And that's using an antibody at saturating conditions. If we lived in a perfect world where we could just dump more antibody in and get a saturated condition with no penalty, that would be awesome. But science is challenging. It is challenging because there's a penalty to every step we take. And the penalty for increasing the antibody concentration is the non-specific signal goes up. And that's depicted right here by this red line. This is just the general haze. If you were looking at this in traditional Western, this would be, if the membrane is just kind of giving signal itself. And that's something that will happen as we increase our antibody concentrations. We will get more non-specific sticking of the antibody to the inner wall of the capillary, no matter how we block that capillary. So our goal is to find saturating conditions where we have a minimal amount of background. And so this is another challenge. Some primary antibodies out there are not amenable to this level of development. And those antibodies are better used for qualitative assays than they are for quantitative. I do have people quantifying antibodies that are not in saturating conditions, but you just have to have that in your head when you're doing your experiments and you're doing your tests from day to day. you might not be getting as good of reproducibility as you potentially could be getting if you were using an antibody at saturating conditions. So it's just something to keep in mind. If you can't get your antibody in saturating conditions, you don't have to throw up your hands and just give up. You know, I would encourage you, because Wes is an antibody screening machine and could potentially screen 12 antibodies at a time, to try different primaries. But if this is the primary that is for that rare protein that nobody else has really looked at, we can do a lot more things with it as well to try and clean up that non-specific signal. But we might just need to be in a non-saturating condition and just have that in our heads that we might see CDs a little bit higher than we would if that antibody was in saturating condition. Now, how do we do this, and how do we do this fast? Typically, and this is when you're working with Angel, this is typically what we would recommend when you're trying out the Wes for the first time and looking at, say, your 2 proteins that you're interested in. Pick a model cell line that expresses this protein. And then we'll do 5-fold serial dilutions of a lysate of this model cell line. And probably start 2 mg per mil, 0.4, 0.08. That's a good range to determine, you know, where we should be working for this lysate concentration. And then, you know, if we're using overexpression cell lysate, 0.25, 0.05, and 0.01 is a really good starting concentration. If you are developing an assay and you need a good positive control, I encourage you all to look at Origin. You know, Origin is not part of our company, but they do have an overexpression lysate and the match control for over 10,000 open reading frames. Pre-made, you can just order it. It's 99 bucks. The lysate comes, you throw it in the system, and it's a positive control and a negative control right there. Really easy way to start doing antibody validation, making sure that your antibody is detecting the right target. So,
Greg, can you put that in the chat? The name of that
Yes.
company? That is terrific news.
yeah, it is one of my go-tos, you know, and I am so jealous that all of you have access to that, because I used to make 293Ts, overexpression 293T cells, or do transient transfection after I clone the protein or clone the, you know, the open reading frame into a vector. And then, yeah, so I would have to do all that myself. And so the fact that you guys can just go and buy it, I am super jealous. You know, that would have made my PhD so much easier if I had access to that. So yeah, it is something. Origin's a great company for that. To be clear, you know, we are part of Biotechne. Novus has those lysates as well, but between us, and this is recorded, so oh well, but Novus sells it for a lot more than Origin. Origin's a lot cheaper. So I usually recommend people just get it from Origin. But anyways, so that is a good positive control. And I actually have a whole online course in our Biotechne Academy on peak validation, which majority of that is controls and how to use them for validating antibodies. It is recorded and available for anyone to use. But anyways, getting back to assay development. Typically when we're doing assay development, we're going to try, you know, at least three different lysate concentrations. You know, it's minimum of 3 points to, you know, see a curve. And these 5-fold serial dilutions are a good way to capture a broad range of lysate concentration. Now, we can combine that with 3 different antibody concentrations, looking for that antibody concentration that's going to be saturating. And if we combine these 2 together, you know, so we have 3 different lysate concentrations, 3 different antibody concentrations. This is where we build our matrix and really in one experiment can test 2 antibodies using this strategy. So in one experiment, 2 antibodies, 3 hours, and we can get and really dial in the amount of antibody we need, as well as the amount of lysate that we need to really get good detection. So typically when you're working with Angel and working with me, and we're starting out from the beginning, this is what we're going to set up. You know, these antibody concentrations can actually be informed by your traditional Western. So if you're using an antibody at concentration X for a traditional Western, we'll typically go 100-fold higher, 20-fold higher, and 4-fold higher as our antibody concentrations. So we don't have to be completely shooting in the dark. We can use the data and all that hard work that you've done for your traditional Western to help us get there fast. But this is where Wes is the antibody screening machine. You know, if you were to try and do 3 different antibody concentrations and all these lysate concentrations and all this cutting up strips on a traditional Western, it would be very cumbersome and very challenging. But for simple Western, this is a lot more direct and a lot easier. So certainly can save you a lot of time and make that transition fast. And this goes to the heart of it. And if there's anything that you remember from this conversation, remember this. Traditional Western, the mindset is, I do the experiment, I look at the results, I plan the next experiment. I do the experiment, I look at the results, I plan the next experiment. And that is because testing multiple things in one experiment on a traditional Western is hard. Testing multiple things on a simple Western experiment is easy. So I encourage anyone who is thinking about Wes to keep that in the back of your mind, that you need to think differently. You need to think about your experiment. Okay, if this happens, what do I want to know next? And put that in the next set of wells. If this happens, what do I want to know next? And put it in the next set of wells. You have 24 different wells on a Wes experiment to do science on. And there should never be an instance where you cannot think of something cool to test in those last 4 wells. If you do this experiment in this setup, this is 22 different capillaries with your negative controls. That gives you 2 other capillaries. And I love sitting with you and designing this experiment and then being like, what else? What else would you just love to see? And then put it on there. You know, you have those wells, you have that ability. It will not affect the rest of your experiment. It will not screw up. You won't have the gel all running all wonky and all that. This is a way that you can test multiple things at once. So when you're planning your Wes experiments, build your if-then tree. You know, plan everything in parallel, not in series. But with that, I would like to thank you again for your time. You know, please, I do highly encourage you to send me an email, set some time with Angel. I would love to sit down, learn about what you're doing. It's always the most enjoyable part of my job is that I get to learn about your cool science and then see what I can do to help you out so that you can transition some of your experiments to Wes and see how it looks. But with that, I'd like to certainly open it up if there are any more questions. I see that Lynette has put the ProteinSimple Academy online for everyone to find in the chat. Again, this is something that I do highly encourage everyone to go through. It is free, so there's a lot of good resources. You will need to look for Wes trainings, but then the workshops are listed under POW, ProteinSimple Online Workshop. But yeah, we're adding new ones to that all the time. I think the series is starting again in a couple weeks. So you can watch them live or you can watch the recordings. I'm slated to do one of them, but I forget what I told them I was going to do. I obviously haven't built it, but that's going to be in December. I'm going to do another one, but I've already recorded 2 of these workshops. I should be recording a 3rd one probably sometime in January. But anyone has any questions, do feel free to take yourself off mute.
So I know that I've had customers who have used very low abundance samples. So I had a customer that was looking at cells in the cochlea of chinchillas, and we were trying to measure a protein in that mixture with treated and untreated samples. And then I've also had customers who have been working with mice, specifically different T cell populations like Tregs, and been able to get good data from those kind of samples where they're, they have to sacrifice quite a number of mice to get, to get the cells, but they were able to reduce the number of mice used per experiment. And yeah, I think, especially like you were saying, Greg, when you have samples that are very precious and you don't have a lot of it, or it costs a lot of money to generate that, then this is a really good system for you to be able to get that expression data that you wouldn't necessarily be able to get with the traditional Western blot. So that's where I see the strength of this system. Plus, it's very quick, it's very fast, it's convenient. You set up your assay and then you run, and then I email you your data and you can analyze it on your own computer after you've been trained how to do that. And I work with you the whole way. I'll help you design your experiments. I set up a spreadsheet so that every time you want to repeat the experiment, you just make another book, and it's right there. It's very easy for you to plan your experiments on your own. And there's a lot of support that I give to all the customers that are using our Wes. And there's multiple Wes's on campus, not just in our core. There's a JESS over in, let's see, I have a name of the unit. And it's on the second floor. Let's see. With research and development, metabolism and translational science core on the second floor of the medical science building. So if you want to have, can you give them a little bit of information about how JESS works in comparison to Wes, which is very similar, but it actually has more, you can do multiplexing a little easier with the JESS?
Mm-hmm. Yeah, so just a little bit about the JESS system. JESS with a J is, JESS is basically Wes 2.0. So JESS has some more features. It has near infrared and infrared wavelengths that we can do detection. So instead of using secondary antibodies with HRP, we can also use secondaries with a near infrared or infrared dye. And so you can multiplex that with the HRP secondary. So you can do multiple proteins, even if they have the exact same molecular weights, all in one capillary. So you get more data per capillary, basically, per sample. And JESS also has a couple of different ways to normalize. So we can do a stripping and reprobing of the capillary once with JESS. And so we can look at your proteins in the capillary and then remove all the amino complexes and label all the proteins with biotin and detect them with streptavidin and HRP. We can do that biotin streptavidin assay on Wes, but it needs to be two independent capillaries. With JESS, it's on the same capillary. So you get a little more bang for your buck on the sample.
So those instruments are on campus, and if you need any more information on how to find, please use me as a resource, and then you have Greg's email as well. And if you have, does anyone have any questions? As I see, there's some potential users here.
Yeah, I'm planning to come see you before too long. The protein we're looking at is also in the cochlea, but we're focusing on the retina. There are a lot of things that we have done with Westerns that have been less than satisfactory, and this looks like it has potential for helping us. Our biggest problem is that this is an extremely low abundance protein, and having enough of it to do things with is the biggest problem. And the other problem is the antibodies. There are a lot of antibodies to this protein, and a great many of them are just not very good. So, but I am sure I will be talking to you, Angel, before long.
So, Dr. McDowell, there is a website called BenchSci. It's
called
free.
what?
Bench, BENCH, Sci, SCI, and they have listed on their website like all antibodies to pretty much almost all antibodies that are on the market right now. And along with those antibodies, they have pictures from the manufacturers, as well as images from publications that are very easy to search. And when if you want, when you come see me, or you can check out the BenchSci website on your own and see if you can't see if there's an antibody there, as well as ProteinSimple has a database that lists some of the antibodies that they offer or that have been tested in their system, in the Wes system, and the concentrations to use them at, and whether or not those experiments were successful. So we might be able to help you identify a better antibody to try.
Okay, good. Thank you very much. I'll be in touch. I only work part-time, but I'll get there sometime.
Yeah, and one more thing I would like to add into that as well is, we're part of Biotechne, and R&D Systems has a lot of antibodies to different proteins, but what they don't have in their catalog, that when you look online, is pretty much every antibody has 4 or 5 sister clones that are just different antibodies that they made to the same peptide. And certainly we've worked with some people when we've really struggled trying to find a primary antibody. We'll contact our colleague at R&D Systems and see if we can find some sister clones that might work for those proteins. And in one case in particular, they were looking at a protein, and there was an N-terminal cleavage, and all the antibodies out there were to that N-terminus, and they wanted to see the C-terminal side of that protein. And we went into that sister clone library and were able to find an antibody that actually worked for the C-terminus. And so it is possible that we can even expand that search. I know that there's already thousands, well, not thousands, but there's probably a lot of primary antibodies out there, but we do have that contact available as well. And we can really try and help you guys find that good primary.
Good, thank you very much.
I put the links in the chat for anyone who's interested in looking for BenchSci and also for the ProteinSimple Antibody Database. One of the things I really love about this system too is the secondary antibodies. You just purchase them directly from ProteinSimple along with their detection system, and they're already pre-diluted, and you just add them directly. It's really fast to add the secondary, and they even have, if you're using a mouse and a rabbit, they have a like 10x rabbit, so you can dilute it if you were to do into the mouse mix, so that you can do multiplexing in the same well. Even with Wes, I've been successful. If the proteins are separated enough by molecular weight, I've been able to do multiplexing. So it's not impossible to do it with Wes. It just takes you have to have all the controls done.
Yeah, we had a postdoc at MD Anderson that worked with us a lot, with my colleague in Texas. developing an assay where she got 5 proteins all in one capillary. And now she's running her lab at Wake Forest. And so I get to work with her, which is a blast because she did a lot of work to get 5 proteins in one capillary, but that's a lot. So it is definitely possible to multiplex on Wes. Do we have any other questions?
So just another comment. So one of the things I think that, especially the engineering students that I've had come through and use the Wes, is the traditional Western blots. It, it takes some finesse. You have to, learn how long you're going to transfer those proteins. And there's a lot of optimization that goes with, and when I say a lot of time, I mean weeks, sometimes months, to get a good Western blot working in the traditional sense. But with Wes, you can literally have an optimized assay in a week. If you have to try different conditions, but because it's so quick, you can run an assay in a day, analyze the data the next day, tweak it, and and it only takes 2 or 3 runs to actually get a really good working assay. So that's one of the other beautiful things about the system. As long as, I think the trick is having a good antibody with this. With any Western blot, but especially I think with Wes, it helps to have a really good antibody and the controls. So I think that Origin website and the Novus, did you say Novus also has the same, right?
Yeah.
Controls? Yeah. Those are.
I think we buy them from Origin and then resell them, but that's just my opinion.
So those controls are really important, especially if you have an antibody that's a little sloppy. Having those controls to know definitely that it's binding to that positive control and not into that negative control. That really makes your data very strong. So I really like working with the system.
And we like working with you.
Well, you just get such really beautiful data once the assay is working right. And then the nice thing is if you're running similar samples, like I did with that chinchilla cochlea experiment, we had 15 animals. Once we got the conditions right, we were able to knock that experiment out in like 2 runs. It was beautiful. And the data looked really good. It was published in Nature Communications. So it's a very good system. So if anyone has any questions, I'll just put my email in the chat too, so you have it. And here's my telephone number. There you go. So please reach out. Using our Wes is very reasonable, and we can talk about all of that offline when you're ready to get started. So Greg, I think since we have all that information up, I think it's okay to quit the meeting now.
Yeah, and I can send you these slides if you would like to have them. And then you
I would definitely like to have the slides. Thank you.
All right.
Yes, me too.
Yeah,
All right,
I conform to you, Dr. McDowell.
Okay, thank you.
Yeah, I'll email them to you at the end, probably at the end of today. I actually got a lot of meetings stacked up, unfortunately.
Right. So you have everybody's emails, right, Greg? When they logged in, or Chris can send that to you. If you
I don't think
register,
Yes,
Sorry.
I have that information. I can pass it on.
thank you, Chris.
Thank you.
Thanks, Chris.
All right, well, thanks everybody for attending today. We're going to go ahead and leave the meeting.
All right.
Nice to see you again, Greg.
Nice to see y'all.
Thank you, everyone.
Weekend.
Thank you.
Yes, thank you too. Bye.
Bye-bye.
Bye.